So STR analysis procedure, usually you will isolate your DNA as always, and then you're going to perform what you call PCR to amplify now the specific loci. And then we are going to also do electrophoresis like we did in the previous profiling. And then of course, once you do electrophoresis, you will do visualization. It can be you used stains, so it is chromogenic. You used fluorescent tags, so you use fluoroscopy, etc. as a method of visualization. So for example, if you have one loci, we are saying that at one loci, you have two alleles, one from the mother and one from the father, because that is a chromosome 1, chromosome 2. So usually you will have, if at this specific point, the STR loci is on both. That's why we call them alleles, the alternate forms of a DNA sequence. So of course you'll have a primer for the first two primers, forward and backward. Then you'll have forward and backward primers for the second allele. So after you have done separation by electrophoresis, you're going to have your two alleles separate. So you have allele 2 there. Allele 2 was longer at 7, so it is going to move slower. And allele 1 was 4 kilobases, so it's going to move farthest. So literally, this is what I have mentioned about how you do or have inheritance patterns, where if, for example, the father has the genotype 36 of a certain STR loci, that is 3 repeats and 6 repeats, the mother has 2 and 8 repeats. Then after meiosis, of course, you know that this will be a haploid, so that there can be now fertilization. So we'll have one having 3, this one 3 and the other one 6, from the mother 2 and 8. And now once they have another generation 2, and there is combination of this, then we are going to have the combinations of 6 2 from this one and this one, and 6 8. Okay, 6 2 and 6 8. And then we are going to have the combination of this other one, which is 3 3 2 and then 3 8. Okay, 3 2 and 3 8. So that is not the second generation. So essentially, these are now the STR loci with their chromosomal positions. At what chromosomal position are we talking about? So usually use around 13 as STR loci. So you can use one of them in any investigation, or you can have them multiplexed. That is where you use more than one of these markers. So we have TPOX, we have B3S1, 358, etc. There are so many FGA. And then we have what differentiates gender, which is called amelogenin marker. This one, amelogenin X and amelogenin Y. Okay. Yeah, so I have put them there. So these are the major STR loci that we usually have. So in short, we are saying we always have to have a specific marker that we are targeting, because even the process of hybridization is using a specific complementary strand. So we must know. So in all the individuals that we are testing, are they 10 samples? Are they 20 samples? We are using TPOX, for example, or we are using TPOX, we are using TH01, we are using VWA at the same time, 4, 3, 10, or even the 13. So the process again is that one, there is a way you can just use one loci. And for example, in this case, you extract the gene TH01 from your sample. And then usually this has the repeating variants of AATG. Then of course you amplify using PCR. You will use primers that will target this region. Then separate by electrophoresis. And then of course run the gel. And then you observe how the separation has occurred of the two or one. Now, if you look at these samples, you have sample A to J. Of course, this is molecular weight ladders. So for sample A, we have 2 separations. For B, we have 2. This we have 2. This we have 2. This we have 1. Remember, these are alleles, the 2 alleles from each parent. 2, 2. Now, for this sample, I want you to note that it has 1, and this one also has 1. What that means is, for example, so we said we must have allele 1 and allele 2, 1 from the mother, 1 from the father. So the one that is 1, it means that it had exactly the same repeats. So if it was 3 for the mother, it is also 3. But the ones that have different means that maybe this is now 2 and this is 10. So the genotype will be 2, 10. But for the one that is 1, it means that it was just exactly the same. The allele was similar. The one that came from the mother and the one that came from the father was exactly the same. Again, now that is when you have the gel as the fingerprint profile. We can also have what we call electropherogram for the STRs. Electropherogram, you have done your primer 1, primer 2, and now it draws for you peaks. So in this case, we had a peak at 4 and a peak at 8. That is 4 and 8. Okay. If you have 6 and 10, the same way, 6 and 10. So this is another way in which you can have the results. Now, that is when you have one STR. Now you can have multiple, what we call multiplex PCR. Now you're having a variety of the STR markers. It's no longer one marker. And of course, this is more sensitive and more discriminatory, particularly in forensic studies. So here you multiplex, or you use more than one marker. Here you can use over 10, the 13 of them that exist. And of course, it's a sensitivity to levels less than 1 nanogram, because you're looking at whichever, you see, when you're having one marker, it's possible that your DNA was not very good quality. So it means that it could miss that particular position where you have fused your primers. But if you're using 10, it means you're targeting so many markers and so many chromosomes at the same time. And that means there's no way you will miss the chance of having a reaction being successful. So of course, it also has the ability to handle mixtures and degraded samples. So in this case, because you're using many, you have to distinguish your markers with different fluorescent dyes. Okay, so that now you can be able to see. That's why you see some are red, green, blue, etc. So usually there are 2 systems that have been there, which we call the 2nd generation multiplex, that was developed in the UK in 1995. It was manufactured by ABI, Applied Biosystems. At that point in 1995, they were using just 6 primers. 6 primers, so 6 STR loci, and that was this one, VWA, the D8, the D21, the D18, the THO, and the FGA, and then the addition of the amelogenin for sex or gender identification. So because of the fact that now we are using more than one, we had to use different, dyes, fluorescent dyes. So we used 5-FAM, JO, and NID. I'll show you a diagram of how the three colors have been used. Now, of course, this process improved into what you call 2nd generation multiplex plus. Still by the Applied Biosystem, but it's the updated version of SGM. And this was around in 1998. And what happened is that they added, now from 6 primer sets, we now have 10 primer sets along with the sex determination loci or markers. And in each pair, each number pair denotes the 2 allele values. That's what we have been saying, the 2 allele values from your mom. So this is an example of some subject or some individual that their alleles have yielded this. And of course, you can see that this particular subject is a female. And the alleles their markers have yielded this allele. So we will now have, if this was supposed to be maybe a comparison, we'll have now the SGM plus profile for another individual, and now we will look at their allele values and we'll be able to tell whether they are related or not, or even in case of forensic investigation, whether there is a relationship between the victim and the perpetrator. So this is an example of a 9 STR amplified with sex typing marker in a single reaction. It's a kit called AMP-FISTA Profiler Plus, okay, by PE Biosystems. So you can see the colors that I was saying: F-farm, blue, Joe green, and then we have mid, which is yellow. And of course, there is the size separation of our gel fragments. So you can see now this, when you do this, you're able now to identify the different markers depending on their color and also on size. Essentially, this is how a genetic analyzer looks like. It has the where you put your samples, the injection electrode, which picks up your sample, puts it into the system. And then, of course, it is using capillary reaction. So there is transportation of DNA depending on size. And then, of course, it will add in the colors, and then there'll be a detection, okay? Of course, there is addition of the polymer, which is now the gel in the capillary system, the outlet buffer. You can see there is inlet and outlet buffer. So this is an automated capillary system of a genetic analyzer that now once you are done, it gives you the results in form of peaks, and it will also give you in terms of the allele numbers. Yeah, so this is a closer look of the sample loading area. So we have the sample vials, we have the capillary system, and we have the electrode that now gets into the sample, picks up the sample, and then puts it into the capillary system. So essentially, you get results like this. Human identity testing now with multiplex STR. Here we have a simultaneous analysis of 10 STRs, including the gender identity STR. So it will yield for you such peaks. You can see these are two individuals. This is individual 1, this is individual 2, and you can see this is now the sex identification marker. Then we have D19, D3, all of them until this one. And the same case for the second individual, all the markers, and you can see they are yielding totally different peaks. So for the amelogenin one, usually if it's a female, it will give you 1 peak. The same way we said, if allele 1 and allele 2 are the same, let's say 3, 3, then you get only 1 band and 1 peak if it's an electropherogram. So in this case, female is XX and this is identical. That's why you get 1 peak. So for the male, you get 2 peaks, that one for X and another one for Y. So you can tell from this one, the amelogenin, which is green, this is XY, and this is just X. So this is XX, okay? So this is a female and this is a male. So essentially, this is how your results will appear when you do a multiplex STR. Mitochondrial DNA, this one, I have discussed it in further details. I'm going to link up the video again up here. So all of a mother's children have the same mitochondrial DNA, which is the same as that of all relatives who share a maternal line. So if we have this generation, this is the maternal lineage. So the mitochondrial DNA in this lady is going to be transferred into all her 5 children. And then in the second generation, if her son marries another lady, then the DNA from the mother, in this case mitochondrial DNA, will not be transmitted because now this is a paternal lineage. So only the new mother is going to transfer now her maternal DNA or mitochondrial DNA. If her child, who is a baby girl, gets now into another generation. Now, of course, we are going to keep propagating the initial mitochondrial DNA that was here into this generation and this new generation. So mitochondrial DNA is a circular DNA that is about 16 kilobase pairs and contains 37 genes, most of which participate in cellular respiration. And it is more sensitive because we don't need so much DNA. And in case DNA has degraded, particularly because nuclear DNA degrades faster, then this is the best method of choice. And when we have DNA that has degraded or it is very old, we're using it on samples that are remains, or maybe it's an archaeological process where you are exhuming bodies that were buried long time ago, or just skeletal remains, hairs that have been found, then we call that low copy number form of DNA. And therefore, mitochondrial DNA analysis is a low copy number DNA analysis method. Of course, we are saying it has disadvantages because if you're in the same maternal line, we cannot distinguish between the two of you or the many of you. Of course, it has more work, more time consuming, and it is quite costly. YSTR is the other analysis method where we get a DNA profile. And this is kind of the opposite of my mitochondrial DNA because this is now only found in males. That is on their Y chromosome. So we investigate a marker, a short tandem marker, short tandem STR that we call YSTR. And of course, we are seeing that these markers are inherited along the male lineage. You can see they're inherited along the male lineage only, and they're highly polymorphic, and therefore they are useful, particularly when there is mixtures and you also want to understand the gender of your biological sample. So here is the Y chromosome. It has quite a lot of markers, 23 markers. Here they are, 23 markers. So this is your Y chromosome. It includes now even the amel Y, amelogenin Y, and all these other markers can be used. These are other short tandem repeats that you can utilize when it comes to identifying if a sample has any male contribution, okay? You want to distinguish, maybe there is a plane crash, which is a mixture of female and male. So before you go so much into looking for their relatives, you first separate the male samples and the female samples by using now such markers of YSTR so that you're able to infer the biological sex. So it can be used in biological sex of a crime scene, yes, to also to exclude male suspects if their DNA is not found. Maybe you have female and male suspects. So if you don't find any YSTR marker positive, then it means there wasn't any male. In population genetics, to highlight multiple male contributors, and then it also assists in paternity or paternal kinship processes. These are other sex typing STR markers that you need to also know. We have amelogenin, we have centromeric alphoid repeats, we have the XF, ZFX, ZFY, zinc finger gene, SRY93, and of course the YSTR. Okay, that's quite a chunk on the DNA profiling methods. Very useful for population genetics, forensic analysis, and any other form of dispute that wants to identify individuals. Thank you so much for being part of this. If you have